Okay, so here we are learning the external anatomy of the renal system. I'd like you to turn to, we kind of looked at renal physiology, and we're going to look at this again. So just as a review, our urinary system consists of 2 kidneys, 2 ureters, a bladder, and a urethra. And if you notice, our blood is filtered through our kidneys. We have, this is a bit of review, we have our 2 kidneys, our right and our left. And notice that the right kidney sets a little bit lower than the left kidney. And if you can see here, you've got your renal arteries, your renal veins. You have your ureters that attach to the kidney, and the ureters run into the bladder, okay, which drains, which drains our urine into the bladder, and then we urinate. We reviewed some of the very important electrolytes that we have here, such as sodium, potassium, calcium, and chloride, and phosphate. And it seems like forever ago that I went over this.
You did.
I know, it's been a while, so this is a good review. Don't forget there's CO2 as well. And of course our blood pH and our glucose play an important role. Our blood pH is always 7.35 to 7.45, and that's really important when we're looking at as the body, as the blood in our system and the fluids become more and more toxic due to the inability of the kidneys to filter our wastes. The pH of the body is going to change. It's going to become more acidic. So the pH is actually going to drop below 7.35. So our kidneys also regulate our volume. They conserve or eliminate water. So if in a healthy functioning kidney, if you have too much water in your system, your renal system will eliminate it. It also regulates our blood pressure, the enzyme renin. So you have your renin-angiotensin system, and we're going to look at meds in a little while. And it also helps adjust the renal resistance in the Bowman's capsule and the glomerulus. Erythropoietin and calcitriol we looked over. Obviously, the erythropoietin helps us build our red blood cells, which we'll look at when we get into hematopoiesis. And of course, calcitriol, which is important in bone formation. And it excretes waste. So that's what our kidneys do. They excrete waste and help clean our blood.
Protein, blood cell factor, vitamin D.
Okay, yep. Vitamin D is important as well. We've got 1 liter of urine that should be excreted per day. And just as a review, this is a really important concept. Most people, for healthy kidney function, should be urinating at least 30 mLs per hour. So roughly around 700 and some mLs per day we should urinate. But that varies if we're dehydrated. It varies if we're drinking a lot. So obviously urinating not enough or too much is bad as well. So we looked at the total renal blood flow, how much the kidney actually filters per day, which is 180 liters, which is remarkable. Now the reason it absorbs 99% of the plasma ultrafiltrate is because within the glomerulus itself, the pores are not big enough to allow plasma proteins to diffuse and be excreted. And the reason for that is we don't want our body to excrete all our plasma proteins because we need them. So the kidney's designed miraculously so it doesn't excrete all the necessary proteins and electrolytes and other elements that we need to regulate our homeostasis. So as you can see, the kidney also regulates blood pressure, synthesizes and excretes hormones. And we said that the nephron is the main functioning unit of the kidney, and that there are about 1 million nephrons per kidney. Now we know that the different kinds of problems that are associated with renal failure. Do you remember what those are?
Renal failure?
Mm-hmm. What are some of the complications that cause renal failure? Do you remember that, Yasmin?
I wrote it down. For the blood vessels and the uterus?
Well, yeah, I'm basically like what can cause the kidneys to go bad? Like we talked about diabetes.
Water.
Yep, diabetes, dehydration, hypertension, kidney stones, nephrolithiasis, renal cysts.
The cysts can kill the kidneys?
Yeah, renal cysts. Yeah, and a lot of people have renal cysts, they don't even realize it. And of course, our kidneys will function, and they'll keep functioning up until the time that only 25% of the kidneys left functioning. And then that's when it really becomes dangerous. So your kidneys will hang in there till the very end. This is a more, I want to say, cross-section of the kidney that helps you kind of see how and where the kidneys are located. So you see that the kidneys are protected. They are posterior to the abdomen and the gastrointestinal system, and that this fatty tissue helps us protect our kidneys from any types of trauma. And so this is the adipose tissue. And of course the renal fascia is the dense irregular connective tissue. So we have what we call a mesentery that holds all of our organs into place. And the mesentery kind of nets everything. So our organs aren't just flopping around in our body. This is the internal anatomy of the kidney. And I drew this on the board last time we were in class, but let's just do a basic review. Each of these little tiny pyramids that you see are basically what we call the renal pyramids. And within each of the renal pyramids, we have a nephron, right? And we have millions and millions of, 1 million nephrons to be exact. And this nephron consists of the glomerulus, which we'll look at in more detail in just a minute. So it's important to understand the renal anatomy. So here you have your outer cortex or your renal cortex right above this dotted line. And then you see the renal pyramids. Now, below that, we have what we call the juxtaglomerular complex, because it's next to it's below the renal cortex, but near the glomerulus. So they sit in there a little bit lower. This is more of a gross anatomy of the interior of the kidney. So you have your minor calyces, where the point of the renal pyramid is pointing to. The papilla are tiny little holes or pores that are at the base of this renal pyramid where the urine drains out. So the urine drains out into the minor calyx, which then is going to drain into the major calyx. So it's just kind of like a tributary of water. If this is a stream, then this is the river. Okay, stream and river. And then from the major calyx, the urine is going to drain out into our renal pelvis, which is this area right here. And that's going to drain into the ureter, which is going to then drain into the bladder. Okay. Now, we're going to take apart each of these nephrons. We're going to look at this nephron, and we're going to look at it in much more detail, in anatomical detail in just a minute. So here we are. So here's our nephron. We said we've got 1 million nephrons per kidney. So let's take a look at the nephron, and let's take a look at the structure. So you have efferent arterioles and efferent arterioles. So remember, efferent is going to, efferent is going away. Afferent going to, efferent traveling away. Now, as you can see, the red indicates freshly oxygenated arterial blood. And that's why it's called an arteriole. And it forms this little tuft of capillaries inside what we call Bowman's capsule. So this little circle here is called Bowman's capsule, where the tuft of the glomerulus capillaries set and filter all of our blood. Okay, what doesn't filter goes back, which is our plasma proteins. And notice that you have this tubular network. So it's important to understand this tubular network. Kind of looks like a nematode here and all convoluted. So as you leave the glomerulus and you leave Bowman's capsule, this is, by the way, where all the pressure builds up and helps the body regulate blood pressure as well. So we move out of Bowman's capsule, or the corpuscle, and we move into what we call the peritubular capillary. And the peritubular capillaries start to descend. Okay, this is what we call the descending loop of Henle. This is the ascending loop of Henle. Okay, so the peritubular capillary, the descending loop of Henle, the ascending loop of Henle, convoluted tubules. So you might look at all of these and think they're all pretty much convoluted, and they are. But each of these tubules where the urine and the waste leaves the Bowman's capsule or the corpuscle go into these tubules and will either diffuse out into the blood to keep, for our system to keep and get reabsorbed, or will be not reabsorbed, but will stay in the tubular area and keep going out to the urine and therefore be excreted. So notice the blue are the veins, and notice how the capillaries and the arterioles are united in this red and blue kind of purplish network. This is how blood is exchanged. So this is how waste products get exchanged. So let's take a look at the nephron itself. So here you see in the left hand corner the actual nephron. And so the renal corpuscle is the site of plasma filtration, where the glomerulus is the small capillary bed. And then the renal tubule consists of the proximal, the loop of Henle, and the distal convoluted tubule, which we looked at in the prior. So these are the renal tubules help us determine which tubule we're talking about when we talk about waste filtration. And of course, I reminded you of the papillary ducts that drain urine from the renal pyramid. So that's really important. So let's take a look at some of the basic segments of the nephron. Now this breaks it down more into the microanatomical diagram, and I'm just going to talk briefly about when the renal system, when kidneys start to fail, this is where all of the action is. This is where kidney failure is going to take place. Okay, so we're looking at the basic segments of the nephron. So let's take a look at this again. So here you see the glomerulus with inside Bowman's capsule. You have your efferent A with an A, arterioles going in, and your efferent coming out. Now it's in the efferent arterioles that we've got these pressure sensors that help determine whether or not the blood pressure is too high. And these pressure sensors, through a negative feedback loop, will work with the kidney to either produce renin that will help raise our blood pressure, or help the kidneys start secreting more fluid if our pressure is too high. And now, barring any health-related disease, our kidneys work pretty good to manage that. But obviously we know with diabetes and hypertension and certain types of renal cysts and chronic UTIs, kidney stones, that eventually the kidneys, particularly here in the glomerulus, take a beating. And if too much pressure is in this particular glomerulus, nothing can get through. I look at it like, for instance, if you have a straw, a tiny straw that you stir coffee with, and you try to pour a pitcher of water through that tiny straw, nothing's going to happen, right? You're lucky if you get a couple drops. because the pressure of the water exceeds the pressure within the actual ability of that little stir to be able to handle that much water. So what happens, everything kind of backs up and stays, right? So if you were to put like a pitcher of water with a tiny little hole, tip it upside down, and put a coffee straw in there, you're lucky if you get a couple drops coming through. And it's kind of this, right? It's kind of the same thing with the glomerulus here. So, and it's within the macula densa itself that we have these cells that help, signal blood pressure changes. So let's take a look. So out from the Bowman's capsule, we go into what we call the convoluted or proximal tubules, okay? And then coming around, we've got the proximal. So we got this, some people will call it the proximal convoluted tubule, so watch how it goes. Kind of twists and like a river or a stream and comes up proximal. We're coming down. This is our descending loop of Henle. And notice some places are thicker than others. This is specific because this indicates a differentiation between absorption capacities and reabsorption capacities of this particular loop. So it's not just by chance that some of these are shaped a little bit different, thicker in some areas, more narrow in others. No, it's because these sites actually function as specific diffusion I want to say factories, where they have a specific job of being able to either hold certain molecules and or proteins and or excrete or absorb. So we've got our descending loop of Henle or Henle, and then we have our ascending loop of Henle. And as we come back up in the ascending loop, notice this line right here is our juxtaglomerular line. So this is your juxtaglomerular complex typically. This is your glomerulus, this is your juxtaglomerulus. So below this line is everything, past of the glomerulus itself. So up we go. Now, because we've come all this way, we are going to look at what happens next. So here we go. Now what we're looking at, now we're up here. Where are we? Okay, so here's the distal. Even though it's kind of like right next to the glomerulus, and you think it's proximal, it's not, because you got to follow the path out through Bowman's capsule. So you get into the convoluted proximal tubules, and you move down. As soon as you pass the juxtaglomerular line, you're now into the loop of Henle, descending loop of Henle, ascending loop of Henle, up to the distal convoluted tubules. And now here we go. Now this, what you see here, kind of like a tree. It looks almost like a stalk of a beanstalk or something, where the leaves would be. This is your cortical collecting tubule. So these collect. They're collecting ducts. Now, similarly, once everything's in the collecting duct, it's pretty much going to go. It's going to be heading down toward the ureters from the collecting duct. If, for some reason, the body says, hey, I need more sodium. I can't discharge that much sodium into the collecting ducts, we need some sodium, then these guys are actually contiguous with one another. It's not as far apart as you see here. We can actually pull sodium out of the collecting ducts back into the ascending loop of Henle. So there's lots of give and take throughout the nephron. So your cortical nephron represents 80 to 85%. So the cortical nephrons are those nephrons that sit at the most exterior of the, I don't want to say they're interior, but they're more distal from the actual renal pelvis. And then, of course, your juxtamedullary nephrons are kind of right on the cusp. So if you take a look, if we go back to this particular video right here, or a handout slide, this is one nephron. Because this nephron is so close to the juxtaglomerular line, this would be considered a juxtaglomerular nephron. The ones that sit higher up, let's just go back here a minute. The ones that sit higher up, like up here, would be your cortical nephrons because they're by the renal cortex. So not all the nephrons lie in a straight line. You may have multiple thousands, hundreds of thousands of nephrons within each of these renal pyramids. And some of them set lower. See what I'm saying? If this is a cortex, some of them set lower. The ones that set lower and closer to the renal pelvis are considered your juxtaglomerular nephrons. And that's why they show you this dotted line here. Okay? So actually the juxtaglomerular are not as widely, there's not as much of them as there are of the cortical nephrons, but they are responsible for creating that medullary concentration gradient to change dilution. So here you have your cortical nephron, which lies a little bit closer. The cortical nephron lies farther away from the, lies closer to the cortex. Okay. Your cortical nephrons. This is much more in depth. This is much more, I want to say, convoluted in the sense that you can see a lot more going on. And remember, all those tiny little red lines here are your arterioles. And arterioles are really important for bringing freshly oxygenated blood into the renal system. Now here's your juxtamedullary nephron. Very close. Lower in the renal cortex. So just like ovaries in a female, once they, develop, they're constant from birth. Any increase in size is an increase in the size of the nephron. So we can't replace our nephrons. It's kind of hard. Just like if someone loses their ovaries, they can't replace their ovaries again. If you injure the nephrons, they stay dead. They can't regenerate. Actually, interestingly enough, brain cells can regenerate. And you can actually build brain cells and increase your brain cells, make new brain cells, but unfortunately not with nephrons. And I'm not sure what type of research is being done at the stem cell level with regard to growing nephrons. I mean, that may be a viable option for people with renal failure, because now they can actually implant ears in people's stomachs, and with the cells, they grow ears on the skin, within the skin of the body, with the implant of the stem cells with ear cells in them, that basically the ear cell,
like human ears.
They can grow them. Yeah, so it's really weird. You'll see somebody with an ear growing under their skin in their stomach, and then they take it out. Yeah, right.
So if someone wants to have a twin,
that's a little bit more complex. That's at a whole nother level. But truthfully, I mean, if they can grow ears, I would think that we're not too far off from being able to grow nephrons. That's a very it's in stem cells, because stem cells are like your pluripotent cells. They're not completely differentiated, so they can still differentiate one way or another into a different type of cell, but given the right impetus, which would be an actual cell from the body, from the human, that possesses these undifferentiated cells, we can actually tell those cells to differentiate, for instance, into a nephron. I think that would be something that might be feasible, although I don't know enough about that particular topic to expound on it. but science is coming a long way. So here we're going to look at the histology of the nephron. So when we talk about histology, we're talking about the tissue that makes up the nephron itself. Cytology would be cells, histology would be tissue. So you have a single layer of epithelial cells that basically form the wall of the entire tube. So these tubes are comprised of epithelial cells. Now you've got to remember, epithelial cells are basically they can be usually single layer cells, not real complex cells. Epithelial cells are everywhere in our body. But some of the distinctive features that distinguish those epithelial cells from, say, our epidermal epithelial cells is that these have microvilli, cuboidal versus simple squamous. Cuboidal is just the shape of the cell, but the shape also dictates certain functions, so that's important to note. But since we're not in cellular biology, we're not going to look at the cuboidal machinery and its dedication to certain types of mechanisms of action. It also contains hormone receptors, which we talked about because, remember, in the kidney, what sits on top of the kidney? What do you remember? The two little hats that sit on top of the kidney. What are those called? Yes, ma'am.
What sits on top of the kidney?
They're called glands.
Glands?
What are the glands called? Adrenal glands. Right? You're my only student in class, so it's like when I put this up on lecture, they're going to say, well, she keeps saying yes, me. She must be like her star student. Well, you are my star student. But you're actually right now the only person here. So it's kind of nice to lecture to someone. So let's come back here and take a look. If I can blow this up a little bit, let's increase the size. So here you've got a little bit of a better idea. Here's inside the proximal convoluted tubule cells. This is the microvilli. Here's the dark little mitochondria. This is kind of like your epithelial cells, parietal layer of the Bowman. These are podocytes. Notice these podocytes look like they have some type of movement ability, which they do. These are like flagellae or flagella, and these podocytes meaning like foot, that allows them to move. Here you have your afferent arteriole, and these are juxtaglomerular cells. And remember, afferent means coming in, where efferent is going away. We're going to look at the macula densa, particularly, you see how the macula densa lies contiguous to the juxtaglomerular cells of the afferent arteriole? This is really important because this is where hormonal regulation of blood pressure is going to occur because of the receptors inside of the, by the macula densa. And so hormones are going to move. This is your cuboidal cellulars. You can see these cells, they look a little bit more meaty, a little bit more thick. This is in the ascending loop of Henle. These are the cells in the distal convoluted tubule. And these are your collecting cells, which consist of these intercalated disc and principal cells. Here's your descending, simple. This looks like simple squamous epithelial. So as I mentioned earlier, you notice the thickness of these tubules, especially you see in the descending and ascending loop of Henle, are not there by chance. It's really important. The thickness is differentiated, of course, by cellular structure. And what we have here is a very intricate network of a structure that is able to secrete, absorb, filter, and help produce necessary hormones to control, control blood pressure and calcium. Excuse me. About 25% of cardiac output goes through the kidney, which is important to know. This is a little bit more anatomy, more of gross anatomy, kind of helping you take a look at how these arteries and veins are broken up. So you see your arcuate arteries, your interlobular arterioles, segmental arteries, and then of course your renal artery. So remember how arteries break down. So typically when we think of arteries, we think of arterioles, and then we think of capillaries, we think of venules, and then we think of veins. So there's this inter-network of a closed system between arteries and veins. I think here it mentions that why the renal circulation is unique, because it's got two distinct capillary beds, glomerular and peritubular, which means that the glomerulus has a bed in which you'll see the podocytes, some of the podocyte cells. It's very important within the glomerulus is that there is a pressure gradient, which you'll see right here. The glomerular capillaries are 60 millimeters of mercury. And here you've got the peritubular capillaries that are about 13. So there's a difference. It's this difference in the millimeters of mercury that cause a directional pressure gradient. I think that might be the best way to describe it. So this kind of breaks down the arteries. I'm not going to test you on all the renal artery breakdown, but notice you go from arteries and arterioles to capillaries. You can go back to arterioles, and basically capillaries are going to go to venules. That's not taking us there. So this is kind of a basic breakdown of the kidney. So now what we're looking at here is the basic kidney process, and it's the sum of these three basic processes that determines the rate of excretion. Okay, so this is kind of a little bit more, I think, structured for you to see. Here's your afferent, here's your efferent, arterial, right? Coming in, going out. Here's your glomerulus. Here's the pressure gradient. Right in here, where the glomerulus and Bowman's capsule kind of meet. This is where that pressure gradient is going to help draw out a lot of the waste products, except for what? What did we say is the one thing that the glomerulus doesn't like to get rid of? Do you remember? What did we say that was? Proteins. Plasma proteins. We don't want to get rid of those. Plasma proteins are too big to fit through the pores of the glomerulus. Okay, so here are your peritubular capillaries. Now, at your peritubular capillaries, let's take a look at what's happening here. Here's Bowman's capsule, and this is your, here we get into your proximal convoluted tubules going this way. But notice what's happening right next to Bowman's capsule as we start going into the proximal convoluted tubules. You've got the peritubular capillaries that are able to either resorb or exchange or diffuse different types of solutes back into the collecting tubes of the capsule. So this is really important, this give and take that you see here at the capillary arterial line. So excretion is equal to filtration minus reabsorption plus secretion. So let's take a look at how the kidney handles some substances here. And this is going to get a little bit more, not to quote, be funny, or punny, it's going to get kind of convoluted. Because filtration isn't that easy. If we could explain it, in the sense that we were explaining it. You had a strainer and you've got spaghetti and you're going to wash the spaghetti and we're going to wash the carbohydrates off the spaghetti noodles. It's understandable. The water becomes milky. We can see a lot of the starch is coming off the spaghetti. The strainer allows the water to come, but it protects the spaghetti from going through the strainer. We understand that. That's relatively easily visualized, but the renal system is not, because there's so many things that are happening simultaneously, and the concentration gradients change throughout the tubules, and then you've got the capillaries sitting right next to all these tubules saying, hey, you need some of this, or okay, give us some of that. So it's kind of like this big Starbucks drive-through, everybody ordering something and some people returning stuff and giving money, and I guess you could think of it as a drive-through because there's so many things happening at that drive-through window, right? So you might think of this as one long drive-through window where things are being given to the cars, and the cars you could think of as you could actually think of the cars as the ducts, as the collecting, as the tubules, whereas the actual Starbucks drive-through are the stationary capillaries and arterioles working, and everybody, you see the hands exchanging money and coffee and whatever. So try to bring it a little closer to help you understand. So here's A. Obviously A and B look the same, but what they're trying to show you here is, look at this arrow. Creatinine waste, where does it go? Boom, right out. Straight through. It's not reabsorbed. Basically the body says, hey, I don't want this. I'm done. Take the creatinine. So if you were to do a urine dipstick, which a lot of, dialysis technicians will do, well, of course, people on dialysis really don't have urine, but if you're working in a urology office or a nephrologist's office, you'll do urine and you'll check for creatinine, because obviously creatinine needs to be in the urine. Obviously, if creatinine's not in the urine, we got a problem, right? What's the problem? If we have to have creatinine in the urine, what do you think's happening if there's no creatinine in the urine? What would you say is happening to the kidney? Is it working properly? No, it's not. It's not, because it's obviously not generating creatinine waste, which means that there's something wrong with the waste processing plant of the kidney itself, which would stem at the glomerular level within the nephron, right? Now look at B, substance, filtration and partial filtration, or filtration and partial reabsorption. So here's B. It's coming down, it's coming down, it's coming down. Oh, some of it gets back into the arterioles, right? This is our drive-through give and change, okay? Oh, here's your coffee, ma'am. Oh, thank you. I actually wanted a double chocolate latte with, 4 sweet and low, an extra pump of vanilla, the drill. That could be your electrolytes, it could be your sodium, it could be your chloride, it could be whatever the body needs at that moment that says, oh, wait a minute here. Can we just have some of that stuff? Can you just give us some of those electrolytes, and it'll diffuse into there? Cool, right? That it has that ability. I think so. All right, let's take a look at C. What do you think's happening here? This is amino acids and glucose. Are they being excreted in C? What's happening to the amino acids and glucose in C? They're not going out, are they? They're not going into the urine. They're immediately taking a right turn at Albuquerque, right? The amino acids and glucose are going back into what? Circulation, right? So remember, this right here, even though it's not draw, I would say let's draw this in red and blues. These are your arterioles, right, that attach to your capillaries. So amino acids and glucose, by coming back into the system, what is that telling you? The body needs them or the body doesn't need them? It's going back into our body. This is a healthy kidney. If it's going back into our body, what is that saying? That we need them.
Yeah.
Right, we need amino acids. Of course we need glucose. Glucose is what? Generated in the liver. If we don't have, say we don't have glucose. The liver generates glucose in two different ways. The liver generates glucose through gluconeogenesis, where the liver takes glycogen stores and turns them into glucose when the body's cells are saying, hey, we need some sugar here. Give me some sugar.
So the person is diabetic. It goes like that?
Yes. Well, eventually, when you look at as diabetics, as their renal system fails, what's really happening, it's the glomerulus is being compromised. And once the glomerulus is compromised, the nephron dies. If you have enough of these glomeruli that get completely bogged down because of too much pressure or too much of a change in pH, which is typically more acidic in diabetics as their sugar goes higher, it kills a lot of the hormonal regulation sensors. So if there's no hormone regulation sensors, everything kind of goes kablooey. Blood pressure goes up, things do, they get diabetic neuropathy because cells can't propagate electronic, synapses and signals, so hormones don't get to do what they're supposed to do. Enzymatic reactions don't go to fruition because the enzymatic activation energy that's required is too high because of the pH that's dropping and becoming too low. And when the pH drops and becomes too low, obviously you don't have that buffer range for enzymatic reaction to take place. Therefore, the enzymes can't lower the activation energy. If the activation energy is too high, it just shuts down, and you get all these minuscule, at the micro level or nano level, all these problems that occur with the diabetic. But the problem is most diabetics don't even know that they have these issues. They think they're fine. They're having a good day, no problems. It's kind of like the kidneys. Once a diabetic they don't take care of themselves, they don't check their sugar, they don't take their oral hypoglycemic meds or their long-acting Lantus or Levemir or insulin, they have their bad days eventually where, I can't get out of bed, I feel like crap, I can't feel my feet feel like there's pins and needles at the bottom of my soles. Why do I feel like this? Well, you feel like this because you haven't checked your sugar in forever, and you're eating bad, and you're ingesting lots of fast-acting carbs and glucose, and so basically your sugar is probably about a 550, 600 right now, and all your enzymatic reactions are being shut down one after another. So yeah, but that takes a minute. Your body's very resilient, and that's why we have to go for regular checkups, particularly if we have family members that have a particular inherited or inheritable disease, kind of like diabetes.
Like that, if someone is diabetic, is it best to see the regular doctor or a diabetic doctor?
Well, it depends. If they're diagnosed, most internal physicians or internists can deal, and they're quite competent and capable of treating people that are diabetic. Oh yeah. Where people go to specialists with diabetes, and when it becomes more complicated, and the diabetes starts to affect the kidneys, then they have to go see a nephrologist, a kidney doctor. If it starts to affect their feet, then they may be, sent a consult for a podiatrist or a foot doctor. The problem with seeing too many multiple doctors is if these physicians don't talk to each other and communicate, then they can start prescribing meds that may be counterproductive to the diabetic itself, for themselves, and actually cause them more harm than good, which is why it's better to have one physician treating a diabetic patient that is still, their condition is manageable and treatable, before, and try to prevent them using preventative medicine to prevent their kidneys from failing. Because that's the biggest thing in diabetics, is we want to preserve their renal health. So it's important that, diabetics, they certainly can see a regular internal physician, internist. That's a good question. So I didn't mean to get off on a tangent here, but obviously amino acids are big. Amino acids are proteins, and we need glucose. So the two reactions in the liver are gluconeogenesis and then, of course, glycogenolysis, which is where the liver will break down carbs once it's done with its glycogen stores. So the liver is really important. Also on the pancreas, the alpha cells of the pancreas will say, hey, we're going to make some glucose. We're going to make sure that the body gets some glucose. And so, you got the alpha cells in the pancreas. So the pancreas and the liver, there's all this hormonal negative feedback. Obviously, it's coming. Also, the central nervous system is implicated in helping preserve this entire system. So let's continue. All right, so these are the blood vessels that surround the nephron. And we kind of looked at this. This is a little bit bigger. This is like your big snapshot. But if you take a look at this drawing, here's your Bowman's capsule. Now, this doesn't look all convoluted. It looks like a little loop. But imagine this is your tuft of capillaries here. This is not drawn to standard. This would be like this big ball of yarn right in here. It's all convoluted. It's a tuft of capillaries, just all kinds of movement going on in the glomerulus, which is why when you measure the glomerular filtrate rate, it's so important. It should be over 60 in men and women. Most of the time, it's between 60 and 90, sometimes 110. When it starts dropping, and I went over this in class, when the glomerular filtrate rate starts dropping, that is indicative that this guy's not working that well anymore. Something's wrong with the pressure gradient. This is dying. These little capillaries are dying. And when they start to die, the glomerular filtrate rate starts to go down. That's not to say that everybody with a low GFR is having renal failure, but definitely if you're dehydrated, this is going to go, your GFR is going to go down. Okay? So your glomerular capillaries are formed between both the afferent and efferent. Well, obviously, because if you take a look at this, we said efferent's coming in, afferent is coming in, efferent's going away. And here you have this, these are all kind of connected together, right? Alrighty. So the peritubular capillaries are juxtaposed or close proximity to the proximal convoluted tubule. And this is really important because a lot happens in the peritubular capillaries. And we're going to see that. So the efferent arteriole give rise to the peritubular capillaries and the vasa recta. And we're not going to get that much in depth for this class. But here is the here's the structure in much more detail where you can say, oh my God, how do I follow that? It's like those mazes that they give you as a kid at IHOP. It's like, follow the maze, come in here, and go out there without running into any dead ends. Remember that? And so this kind of breaks it down. I'm not going to go over everything because we kind of reviewed this, but this is kind of in its entirety if you're viewing the nephron. This is it. There you go, Mr. Nephron. And here's the papillary. These are the little holes where urine drains out. This is kind of the flow. So you've got your glomerulus, and goes to Bowman's capsule, goes to the proximal convoluted tubule, to the descending loop of Henle, up the ascending loop of Henle, through the distal convoluted tubule, and to the collecting ducts. And here's our kidney, a little bit of a side-to-side juxtaposed structures, your ureters, kidneys, renal arteries, renal veins. Remember, veins go to the heart, arteries go away from the heart. So veins are going to. So arteries are going in, veins are going out. Bladder, Okay, there's your bladder. Now the renal corpuscle is the first part of the nephron, and the afferent arteriole leads to the glomerulus, we know that, which is a specialized capillary bed. And we're going to do one more sentence here. We're going to take a short break. This is what I was talking about with respect to pressure. Filtration and filtrate. What's the difference? Filtration is based upon what? The difference in hydrostatic pressure in millimeters of mercury between the glomerulus itself and the space in Bowman's capsule. There's that pressure gradient that kind of helps pull everything out from the glomerulus and drain into Bowman's capsule, which is your filtration. Filtrate is what is the fluid. Filtrate is the fluid, and it consists of everything except blood cells and large proteins. So here's my question to you again. Why don't we just filter out into our urine all our blood and proteins? How long would we last if that happened?
Not long.
Not long, no. We'd be having hematuria, which is, of course, blood in the urine. And we'd be losing large amounts of protein, so yeah, we wouldn't last long. Not too long at all. So the body preserves that, okay? So let's take a short break, and we shall return. So we had a little short break, and we're going to look at, now, we're just going to try to finish off the renal corpuscle. The corpuscle of the corpuscle, this is it. This is where you can see what's happening. How closely the macula densa cells are contiguous with these juxtaglomerular cells. So here's your, this is kind of upside down, I guess. This is efferent going away, afferent going to. Here's your glomerulus. Here's Bowman's capsule. So you can see that they've blown this up, and you see this space, and there's pressure gradient here. There's a pressure gradient here. Here's your proximal tubule. Now the podocytes, those little walking guys, this is inside the visceral layer of Bowman's capsule, and it's basically on the glomerulus itself, or the visceral layer would be that particular layer that's almost like mesentery that adheres to the actual structures. A visceral layer usually contains fluid as well.
Should we get text ID number? It's doing this call.
And so if we take a look, the arterioles themselves have these juxtaglomerular cells next to them. This is the endothelium. And endothelium, when they say endothelium, it usually is indicative of the type of cell. Notice these cells are like simple squamous epithelium or epithelial cells, whereas these cells seem a little bit more specialized. They've got the little microvilli on the surface to help propel in a particular direction. Okay, so villi is important. So as we know that the glomerulus is the primary filtering device of the nephron, the capsule of Bowman's capsule itself consists of a visceral and parietal. And those little podocytes or walking cells are a differentiation of squamous epithelial. The parietal also consists of squamous epithelial and forms the outer wall of the capsule. And the capsular space is basically that space where that pressure gradient exists between the glomerulus itself and the 60 and the 13 millimeters of mercury. So here in more detail is a structure of the renal corpuscle. This would be the external view, so you can't really see what's going on. Slice this open and you think, oh, well, there's everything, right? Here it is. So this kind of helps you understand how I want to say dense the glomerulus actually is and how specialized it is. And all these specialized cells that we've talked about, we're going to see shortly that your cells of the macula densa are implicated in controlling of blood pressure. So here we go. Here's the filtration membrane. So we're looking at even a close-up of the actual glomerulus itself, and it's like, wow, look at that fenestrated layer. There's our sieve. Here's our spaghetti sieve that we talked about, right? And then you have layers. You have a filtration slit. You have your pedicle, your podocyte of the visceral layer of Bowman's capsule. Here we go. Even closer look. So obviously these pores are small enough to filter water, but too small to allow proteins, plasma proteins and glucose. Negative charges block the passages of protein. So if it's a negative charge, proteins would be positively charged and are also negatively charged. If it was positively charged, it would not block them. But so similar charges will be blocked. Also, charges themselves, which you can say negative or positive, are also specific to the type of activity of a particular structure. So charges are really important. Neutral proteins that have no charge won't be repelled or attracted. So if there's no charge, it's not going to go through either. So here's your specialized cells. Now these JGA cells are important. This is where we're going to see that the blood pressure is regulated. Most of these cells of the distal convoluted tubule are your principal cells, which have receptors for the ADH, and aldosterone. This ADH is your antidiuretic hormone. When we talk about antidiuretic, what are we saying? What are we actually saying? What's a diuretic? What does a diuretic make you do if you take it? A diuretic. True, but the actual purpose of an antidiuretic is to keep you from urinating, right? So yeah, it will increase your blood pressure. It absolutely will, as will aldosterone. And the intercalated cells also help maintain blood pH. So again, we see this Bowman's capsule and glomerulus. Where the macula densa is a thickened part of the ascending limb and the juxtaglomerular cells are modified in the arteriole themselves, which regulate blood pressure. These cells are specific to hormonal regulation and pressure. So when the juxtaglomerular cells are activated or infringed upon, they're going to react. And it's a big chain reaction that takes place going back to the pituitary gland and the hypothalamus, which also controls blood pressure through the secretion of different hormones. So the three processes of urine filtration we looked at. This is kind of a review. As this is a review, and these are just some of the layers. Diameters obviously differ, just like we saw in the descending loop of Henle and the ascending loop of Henle. Difference in diameters will cause a difference in pressure and a difference in pressure gradient, along with the specialized cells. So I'll let you read this. Here's a summary of the forces. So you've got your efferent coming in. Here's your hydrostatic pressure of 60. Here's your colloid osmotic pressure of 32, which basically is saying this is not going to go through. These are your proteins, your colloid osmotic. Then you've got Bowman's pressure. Okay, so this pressure can be affected by multiple factors. As you were saying, Yasmin, also with the when the blood pressure is too high, the diffusion capacities are going to be altered on the membrane permeability to help regulate water and help us, urinate. So you've got a net filtration pressure of 10. Now this hydrostatic pressure of 60, it may or may not be associated with your GFR rate, because GFR is not just pressure. The glomerular filtrate rate, because rate is part of the units, is in distance per time, which is velocity. And we're also measuring pressure, and we're also measuring the diameter of the tubule that it's going through. So there's multiple units associated with the pressure gradient. So this kind of goes over the resistance factors, and how the GFR rate is affected with renal blood flow. Your renal blood flow decreases, your pressure decreases, your GFR decreases. Dehydration right here. This is dehydration, or too much water loss, too much fluid loss. People on dialysis, well, obviously they don't have, their GFR is actually non-consequential, but in someone that's dehydrated, this is what you'd see. A decrease in renal blood flow here, an increase in the pressure gradient increases. This should actually be an increase, not a decrease. I'm not mistaken. Constriction of the efferent arterioles increases the resistance. So I guess here, what's going to I see what they're saying. So basically, if this resistance goes up, then it's going to push back. So if this is, basically this is the corrective measure of our body. When this is low and you're dehydrated, then your efferent or efferent going away, arterioles are going to constrict to push back whatever fluid is going out, so that the pressure will go up and the GFR will go up. This will evidently raise your blood pressure. So basically you see this, and this is the corrective measure. So here's our little tuft of spaghetti here. Your efferent, your efferent, your capillaries, your Bowman's capsule, your GFR. Here's your reabsorption, right, of plasma proteins and glucose at the proximal convoluted tubule into the peritubular capillaries. Interesting right here, I just want to, before we finish this PowerPoint slide and we'll go on to the next one, is that 99% of all the fluid filtered is reabsorbed. That's pretty amazing. It's reabsorbed. That means we need it. We're only 1%, is your waste. So that's a pretty remarkable machinery. Again, these particular fenestrations stop your cells and platelets, stops your plasma proteins, and medium-sized proteins, but not small ones that have been broken down that you no longer need. So this basically protects us from losing all of the specialized cells that we need, our blood cells, etc. So it's the fenestrations within the GFR. Okay. So this is glomerular filtration rate, and it's not just mLs per minute. This filtration rate, if you look it up, it's got a lot of different units in it. It's volume per liter, it's distance, it's in distance, it's in velocity. So So homeostasis of the body requires that our glomerular filtrate, it remains constant and it doesn't fluctuate too much. But obviously, if we exercise a lot, we don't drink enough fluids, our GFR rate's going to go down. If you lose too much weight and you don't eat properly, your GFR will go down. Those are just like people that are anorexic. You've heard of anorexia. They become very cachectic, where they lose a lot of their body mass and proteins. And basically, the kidneys can't keep up with it, and they get bogged down with too many plasma proteins and not enough fluid. And in many cases, people that are what do you call it? They don't. They're what did I just say? They don't want to eat. They're anorexic. They can die of kidney failure. Their actual organs begin to shut down. Net filtration. So the three main pressures are your osmotic colloid, your glomerular filtrate pressure, your Bowman's capsule pressure, and you've got a net filtration rate of 10 millimeters of mercury. Not that that's important. This is really important right now for us, just to understand what happens, how the kidney functions, and then of course why people that are on dialysis, why their kidneys fail and why we can't bring them back. Obviously we can't regrow nephrons. Nephrons don't replace themselves. So once the GFR rate drops below 15, and 15 is not always the cutoff number. I mean, I have seen people at 15 that still were not candidates for dialysis, because we can bring up our own GFR if we can drink fluids, and because we don't just look at GFR, we look at a lot of the electrolyte concentrations in the body to see, particularly what are we looking for in the urine? Creatinine, right? Because the urine has to, the kidneys have to what? Get rid of creatinine. So your glomerular filtrate rate is regulated by not only your sympathetic nervous system, but also your parasympathetic. Now this is where your hormonal factors will actually affect regulation of your GFR. Angiotensin 2 is a product of your renin-angiotensin pathway. So obviously, if the kidneys produce renin, renin is going to combine with angiotensin 1 to form angiotensin 2, which will eventually form vasopressin and raise the hydrostatic pressure. Endothelial-derived nitric oxide decreases vascular resistance and increases the GFR, which means you will actually urinate more. Prostaglandins and bradykinin decrease vascular resistance and also increase the GFR. Here we go. Once again, we see it in a little bit different drawing. You see the different specialized cells. You see the glomerulus, the epithelium, the afferent and efferent arterioles. And this is the macula densa cells that we talked about in the juxtaglomerular cells in the complex next to the afferent arterioles. It's the sodium chloride that can cause dilation of these, which can therefore raise the glomerular hydrostatic pressure. And as you can see, this is where your electrolytes become important, which is why they say where sodium goes, water follows. The more sodium is taken up, the higher your blood pressure, and it'll create this cascade. This cascade. Now, this cascade is to help raise your GFR. So if there's a decrease in GFR, then there has to be an increase in sodium chloride absorption, which will cause an increase in water, which will then cause an increase in renin and angiotensin, which will increase your blood pressure. And so you have your regulation of fluid waste. It's the stretch in the afferent arterial smooth muscle that causes these cells to become, I want to say, excited to do what they're supposed to do, whether it's absorb sodium or release sodium to lower the GFR. So if you have elevated BP, then the tubules, sodium and chloride, are not going to be reabsorbed. Water will not follow. And the constriction of their arterioles are going to reduce the GFR so that our blood pressure will drop. Because when one constricts, the other opens. So if efferent constricts, efferent's open, and therefore reducing the GFR. So all this auto-regulation is done through hormonal feedback, through specialized cells. Here's your neural regulation. Here's your hormonal. Now you talked about vitamin D, right? Your calcitriol. Antidiuretic hormone, atrial natriuretic peptide, sympathetic stimulation, which is not hormonal, but these are all hormonal. Your ANP, atrial natriuretic peptide, increases your GFR, stretching the atria, causing an increase in blood volume, increasing the capillary surface for more filtration, thus increasing the GFR. High blood pressure obviously reduces the GFR, right? Low blood pressure and well, I shouldn't say that, because we have to think about how your body is going to fix the problem. So let's just say it this way. If your body wants to get rid of water, you're going to increase the GFR. If your body wants to retain it and raise your blood pressure, you want to reduce the GFR. So think about what's happening with the body and what you want to happen with the body to help the body regain or maintain homeostasis, and that will tell you what you want the GFR to do. So if you're really dehydrated and you don't have a lot of fluid, your GFR obviously is going to drop because it doesn't have enough fluid to push through. You're changing the pressure, so we want to increase the GFR, and to do that we have to give people fluid if their body is normally functioning. Obviously, your antidiuretic hormone stimulates the aquaporin 2, where water molecules move more rapidly, and you get, it's stimulated by a decrease in blood volume or severe dehydration. So your body's going to try to say, hey, we're really dehydrated here. I'm going to release a lot of this antidiuretic hormone so that it constrict the efferent arterioles, push it back, and we can start stimulating blood pressure to go up and water and conserve water in the body. So once again, we take a look at urine formation. What stays, what goes? Creatinine waste goes, amino acids stay, electrolytes stay, organic bases and acids, some stay, some go. So it just depends. This is the reabsorption process. As I said, the peritubular capillaries and the renal tubules are next to one another. So you've got this exchange. This is your Tim Hortons, or I think I use the term Starbucks drive-through, right? Exchange of ions and things like that. What stays, what goes, plasma proteins, etc. So this kind of tells you what's happening, what's going, what's coming, what's absorbing. And of course, we talk about active transport. Now, active transport means that there has to be energy put into the system for this to take place. So when I talk about active transport, I like to use the analogy of an Uber driver. Okay, active transport. So the person or the passenger is who the Uber driver is transporting, and the car is the mechanism of transport. So you need to, and in many cases, he's got to put on the gas to go. That's your adenosine triphosphate, or your energy molecule. And the car is the actual molecule that's going, that's holding the passenger, which is the solute, that needs to be transported to either to go outside to the urine or back inside the body. Excuse me, if it has to go back inside the body, it needs an Uber driver. I think that kind of makes sense. ATP, adenosine triphosphate, is the gas in the car of the Uber driver. He's not going to go anywhere without gas, right? And he can't transport his person back into the peritubular capillary, into the body, unless he's got gas in his car, which is your ATP, and he's got the passenger, which is the sodium, potassium, chloride, CO2, plasma protein. Typically, your bigger molecules have to be transported via ATP because they're too big to be just diffused. Okay, secretion is usually, remember, take secretion, small molecules are secreted, charged molecules are secreted. So this little arrow, if I blow this up, shows you what's coming and going. So, you know, organic acids and bases and hydrogen, boom, we are sending that down the tubes out to the urine, whereas your sodium, chloride, bicarb, potassium, water, glucose, and amino acid proteins are going back into the body. So these arrows can be a little bit, you know, hard to read. This means boom to the urine. This means boom back into the blood. So I'll let you take a look at that. This is how urine is formed. Kind of cool. We're going to take a look at the components of urine on the urine dipstick vials. It's kind of cool. The different types of compounds that are in urine. If you want to do this, let's just take a look real quick. Let's Google, since I don't have the book open. And let's look at components of urinary dipstick. I'll probably pull up some type of lab. So let's just do this. I like Wiki, but you gotta stay away from Wiki. Let's take a look at this. Let's see if we can open this. Some of these are not, you can't open them. Okay, so because these are litmus paper and they are, when you dip it in the urine, the litmus paper reacts accordingly. I'm trying to see.
Like if we check if the patient has infection?
Right. So leukocytes are your white blood cells. And
yeah, this sticks. It has like lines to it.
Yeah, you've done this before, right? So it tells you leukocyte esterase. So if you have white cells, you have, you know, an infection. Nitrates are also the byproduct of bacteria. Typically gram-negative. Greater than a 10,000 bacterial colony count, they typically say that you have a contaminated specimen. Someone contaminated it. All right, let's continue. Hematuria obviously are red blood cells. There shouldn't be red blood cells in the urine. Glucose, you know, you're not supposed to have a lot of glucose in the urine. Different things can cause that. Typically, if you find too much glucose in the urine, it's usually indicative of diabetes. Ketones, well, this is a result of diabetic ketoacidosis. Ketones you really shouldn't have. Ketones are also, high levels of ketones can come from bodybuilders as well as proteins. Bilirubin, Not usually present in healthy patients, but obviously if there's liver disease, bilirubin will be found. So those are just a few. Let's go back. Urea. There's 10 altogether, actually. And this goes over urine analysis. And it tells you kind of like the color. So when someone's collecting, here's your bilirubin, your protein, your pH, your specific gravity. Almost a healthy urine, the specific gravity is almost like water. It's not too much heavier than water. If something's cloudy, that means the turbidity. Obviously, if we know what urine looks like, if it's not healthy, we can see that it could be cloudy, it could have specks, it could be red tinged.
What does it mean, like, if the urine, like the poop stool, if it's black?
If what's black?
The poop?
Oh, feces, stool. Stool is black? Well, it could mean a couple of things. If someone is black stool, typically, if the patient is taking iron pills, their stool is going to be really black and tarry. If they're not, I would have that person see a physician because that's sometimes a sign of internal bleeding. Right. You also want to look for blood in the toilet as well. So we're going to stop here. All right. And